Viral replication is when a virus needs to go through a process of replication to create new infection virions that are able to infect other cells of the body. When they gain entry into the body, a virus makes its physical contact and crosses the plasma membrane or the host cell. Then it releases and replicates its genome while making it possible to make its proteins by host ribosomes. Then the virus particles are assembled from the new synthesized biological molecules and become infectious virion. Then the virions are released from the cell to continue the process of the infection all over again. The Lytic cycle is a 6-stage cycle which are attachment, penetration, transcription, biosynthesis, maturation, and lysis. Attachment is the first stage, when the phage attaches itself to the surface of the host cell in order to inject DNA into the cell. Then the phage injects its DNA into the cells by penetrating through the cell membrane. Host cell's DNA is degraded and cell metabolism is directed to initiate start phage biosynthesis. The phage DNA replicates inside the cell, synthesizing new phage DNA and proteins. It replicates material, assembles into fully formed viral phages, and then the lysis stage comes into hand. Cell wall ruptures and breaks up the virus in the process. The end.
I will be explaining the lysogenic cycle. We'll be starting from up here and then working our way around. And the first part of the lysogenic cycle is after the virus injects its own DNA into the cell. And then after that, the DNA is actually going to become fused with the host's DNA. And then the cell will then replicate the DNA and then go through binary fission, as each new cell is going to have the viral DNA. And then as more and more cells duplicate, there's going to be more and more viral DNA. And this viral DNA will eventually go out and produce viruses that will go and infect other cells. And that's the lysogenic cycle.